1999 from University of Peradeniya in Sri Lanka, and he obtained his master's in marine biotechnology in 2007 and the PhD in aquatic life medicine in 2010 from the Jeju National University, South Korea. After he obtained his postdoctoral research at the Department of Marine Medical Sciences in University of Jeju, and he joined Chunnam National University and established the Aquatic Animal Health Laboratory at the College of Veterinary Medicine. And he is interested with his research and spans diverse areas related to prevention and control of multi-drug resistant pathogens using antimicrobial peptides, nanomaterials, phage therapy, and development of disease models in zebrafish. And Dr. Mahanama has received the highest honors research achievement award from the president of the Jeju National University in 2010. And he is also the sectional head of fisheries and aquatic sciences at the Department of the College of Veterinary Medicine, Chunnam National University. And he also has obtained more than 125 peer-reviewed international journal publications and more than 4 book chapters. And under him, more than 2,500 citations and H-index also around 29. So the topic today, the Professor Mahanama has been chosen, the antimicrobial peptides against multi-drug resistant bacteria. I think now the time to start the webinar, and any questions to receive to speaker, please send it to the chat box. Thank you. Over to you, sir.
Okay, thank you very much, Dr. Kalpa, and good evening to all of you. First, I would like to thank Dr. Kalpa for inviting me. Actually, he invited several months ago, but due to some work, I had to postpone. So basically, I think Kalpa gave all the information about me, so I'm not going to repeat. So I have selected one title because of our ongoing work related to antimicrobial peptides. So I'm going to briefly discuss about the current status of antimicrobial resistance, and also next about the antimicrobial peptides and clinical applications. And last part, actually, I want to summarize related to our studies or our research with some of the novel antimicrobial peptides which we are working on. So as a introduction, actually, I searched the key events related to the antimicrobial resistance as a timeline. So if we go to the 1900s, so we consider as a pre-antibiotic era, and then antibiotics were discovered. So that period is considered as a golden age of antibiotics. And there is another short era called the Me Too drugs. I'll explain you later about that. And then we are at the moment at the discovery void stage, or the era related to the antibiotics. So very recently there was one conference actually in 2019, and they discussed about the key issues related to this antimicrobial resistance. And then one of the keynote speaker, Professor Doug Bird, he mentioned this as a climate crisis of medicine. So we are all discussing about the climate change and issues related to environment. So they are highlight this issue as a climate crisis of medicine. So let's little bit talk about this pre-antibiotic era. So before discover the all antibiotics, actually the most of the death uh, is related to the several key diseases. So the flu or the gastrointestinal infections like diseases. But once the antibiotics were developed, basically the first discovery of penicillin by the Alexander Fleming, and then it started to develop many new antibiotics. So you can see in 1945, the Alexander Fleming received the Nobel Prize. So during his the awarding ceremony and his speech, he has warned about the penicillin resistance. So it is just after several years of starting that warning, the people realized that and found in 1948 the penicillin resistant staphylococcus. So that time it was announced as a global pandemic. So after that, usually what we do when we fail something, we try to find some alternate materials. So what the people tried actually at that time, they tried to use other antibiotics to treat the penicillin resistant staphylococcus species. So because at that time there were a lot of new antibiotics, so especially between 1950 to 1970, more than 20 new antibiotic classes were discovered. So soon after that, it was revealed that methicillin resistant staphylococcus also documented in worldwide. So at that time, what the scientists tried to do was they try to modify available antibiotics slightly, and they try with the different uh, resistant pathogens. So that was another issue, and it caused more and more resistant problems among the available or existing bacteria, especially. So in 2001, World Health Organization, they declares the antimicrobial resistance as a global public health issue. So after that, there was a key incident in 2013. They explained that a lot of antibiotics have been used in agriculture. So it can be easily move into the other animals and then um, come back to the human, and then this situation become more and more serious. And in 2015, the first report of colistin resistant MCR1 gene was discovered. So colistin is one of the most strongest uh, antibiotic which used to control the highest resistant strains. And also in 2017, the Klebsiella pneumonia strain resistant to uh, those antibiotics. So there is one study actually they have predicted in 2050 there will be uh, 10 million deaths per year, and it costs a huge amount of money which is related to this antimicrobial resistance. So now we are in a real crisis related to this antimicrobial resistance. So every country, they have, most of the countries, actually developed countries, they have started their specific strategies to overcome this problem. Uh, so here, there are several species related to this resistance we have to concern. So one set of bacterial species we call escape pathogens. So there are 6 pathogens are grouped and which are in the priority list related to this antimicrobial resistance. So they are the Enterococcus faecium and Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter species. So here in this graph, you can see the level of the antibiotic resistance. So mostly in this study, they proved uh, almost all these listed pathogens are highly resistant against the commercially available antibiotics, right? So generally they develop various mechanisms to escape from these drugs. So when we think about this uh, resistance, then we have to focus on how they develop these mechanisms. Why it is important? Because if you try to develop some alternative materials against these uh, pathogens, so then it will be very useful to find out some targets to produce these new materials. So there are 4 major mechanisms have been discussed related to these escape pathogens. First is related to the enzymatic inactivation of the uh, antibiotics. So these pathogens are able to produce certain enzymes like beta lactamase. So these enzymes are able to change or modify the active sites of these antibiotics. So for example, so this beta lactamase can uh, permanently or temporarily can modify the beta lactam ring in these antibiotics. So therefore their efficacy can be lower. At the meantime, they are able to modify the amine or the hydroxyl modification, right? So this type of modification can lead to have the very low efficacy uh, antibiotics, and therefore the bacteria may not inhibit properly. Then the other strategy is related to the bacteria. So as you know, they have the specific pathogen associated molecular patterns like a lipopolysaccharide or the peptidoglycans like that. So they are able to modify the target sites of these antibiotics. So thereby, so they can escape from the effect of these antibiotics. And the other strategy is to uh, makes the persistent cells while forming the biofilm formation. In the case of biofilm, so these planktonic bacteria, they attach to the certain surface. It can be the some dialysis tube or any other medical equipments. So then they produce these extra cellular materials and it mature in a specific area, and then some of these cells can be moved to the escape and move into the environment, and these other cells, persistent cells, can form a specific uh, area which are very resistant, strongly resistant to the antibiotics, right? So other one is reduce the accumulation uh, of the antibiotics in the cellular environment. So especially it is related to the cell membrane functions. So this porine alteration can reduce the intake of antibiotic into the cell, or it block the or it remove the antibiotics from the cell. Therefore, these bacteria, especially these resistant bacteria, uh, uh, having different type of strategies to overcome the antibiotics, then they can develop the resistance. So to give you some idea about the real situation, this antimicrobial resistance in our day-to-day life, how it affects to us, because it's highly connected with various environmental or our agricultural activities, especially it comes into the food chain. So here you can see when antimicrobials are given to the food producing animals for their diseases or sometimes as growth promoters, and then they develop the resistance, and then it comes into the environment and the food, and thereby the human can have this type of resistant bacteria. And then once they are not healthy, and when we have to treat these treat those infected human or the people, then the resistance can cause problems related to the low efficacy of antimicrobials. So therefore, we have to focus on alternative strategies to overcome this antimicrobial resistance. There are many alternative materials, natural products, bacteria, the materials, antibacterial materials. So here I am focusing on use of antimicrobial peptides as one of the good candidate to control these antimicrobial resistant pathogens. So let's talk about what are antimicrobial peptides. So these are some of the basic characteristics of antimicrobial peptides. So they are generally called the host defense peptides. So normally in naturally, actually they are gene encoded uh, antimicrobial peptides. All the living organisms, they have specific genes which related to the antimicrobial peptides, or sometimes in mostly like bacteria, they have a non-ribosomal antimicrobial substances or peptides. So usually when we define these peptides, we consider they are short chain peptides, generally uh, 10 to 15 amino acids, 10 to 50 amino acids, and mostly they are cationic. So they have a net positive charge between +2 to +11. And based on their secondary structure, we can categorize these antimicrobial peptides into 4 categories: alpha helical structure, beta sheet, extended coil, or cyclic. So there is a highly diverse structural related antimicrobial peptides in our natural sources. And then another important thing is the amphiphilic nature. So they have a hydrophilic and hydrophobic amino acids. Generally, this hydrophobic feature is very important to penetrate in the lipid membranes. And here in cationic and antimicrobial peptides, it's very important to make the electrostatic interaction between the negatively charged cell membranes, especially in the gram-negative bacteria. So functionally, these antimicrobial peptides have broad spectrum of activities, antimicrobial such as antibacterial, antiviral, so on, and also antibiofilm activity or anti-cancer or anti-tumor activity, anti-inflammatory activity or wound healing, immune modulatory activities. So it's not just antibacterial or antimicrobial. So the single peptide may used for various other therapeutic applications. So that's the highlight of this antimicrobial peptides. So these are some of the examples with the 3D structure of the naturally available antimicrobial peptides. So here catholicidine or manganine, this is from the frog, is related to the human, and also the defensine. So defensine is one of the main antimicrobial peptides we can find in plants and also invertebrates, vertebrates, and all kind of animals. So there are various isoforms of defensine. So this melitine and defensine in tomato, like that, right? So this is just to give you some idea the different structural of the antimicrobial peptides. So when we compare the mechanisms or the targets of antimicrobial peptides and antibiotics, we can find several similarities between those. So in here left side you can see the antibiotics, in the right side antimicrobial peptides. So basically if you compare the antibiotics, they are mostly inhibit the cell wall synthesis, and also they inhibit the ribosomal synthesis, different type of ribosomes, and also they are able to inhibit the nucleic acids or the DNA synthesis. So these are some of the examples which with the known mode of actions of those antibiotics. So on the other hand, if we consider the antimicrobial peptides. So most of the antimicrobial peptides are functionally, or their mode of action is related to the membrane disruption. So usually the positive charge is related with this membrane disruption, and then it changed the membrane permeability, and then it caused the lysis of the cells. So you can see most of these antimicrobial peptides are characterized with that function. So and also some of the antimicrobial peptides are associated with the inhibition of the DNA or nucleic acids, or the inhibit the cell wall synthesis. So you can see some of the similar functions between related antibiotics and the antimicrobial peptides. But additionally, you can see some of the antimicrobial peptides are able to modulate the immune responses. It is not in the host, it is in the not in the bacteria, it is in the cell. For example, when you treat the antimicrobial peptides like defensine, so it can induce the neutrophils to produce the pro-inflammatory cytokines, and then it activates the immune responses towards the better immune function in the host cell. So altogether it can help activation of the overall immune system and make the control of the pathogen. So let's briefly talk about this discovery and the clinical application of antimicrobial peptides. So you can find the information about the antimicrobial peptides. There are various type of databases. So this is one of the database. So you can get the more than 4,000, almost 5,000 antimicrobial peptides. Most of them are natural, and there are around 70 peptides are as a target drug candidates. According to this reference, it mentioned about 34 peptides are under the preclinical stage, and the 27 and under the clinical stage. And there are 7 FDA approved antimicrobial peptides are being used. And these are the 7 FDA approved antimicrobial peptides and their structures. So when we consider the mode of action of these currently testing for the clinical, so there are 3 different mode of actions they have classified. One is the direct and antimicrobial activity via the cell membrane. As I mentioned earlier, most of them are change the cell membrane potential and then cause the cell death. And then some of those peptides are indirectly cause the antimicrobial activity via the immune modulation. Others are related to the inhibition of intracellular functions like uh, for example, suppress the expression of the specific proteins important for the cell wall synthesis like that. So when we consider the importance of antimicrobial peptides as a therapeutic agents, there are several benefits we can consider over the antibiotics. So one of those are rapid action on the distinct biological targets. So when we use the antibiotics, especially, we may not get the clear idea whether these antibiotics can kill the beneficial bacteria. But here mostly these antibiotics are relatively specific than the antimicrobial peptides are relatively specific and their action is relatively fast. And mostly reports say that the development of the resistance is relatively slow for the antimicrobial peptides, and also the less toxic to the eukaryotic cells. So in the safety wise, it is more reliable way to treat. And also to avoid the misuse or overuse of antibiotics, we can combine the antimicrobial peptides with antibiotics. So thereby we can support for reducing the development of the antibiotic resistant species. Other than that, these peptides have various type of biological functions or the applications like wound healing, immunomodulation, so on. So next part, I want to explain some of our research work. So this is our strategy. We uh, focus on our project to develop antimicrobial peptides. So what we have done actually, we use one marine animal, the octopus minor. So we did the transcriptome sequencing of this octopus minor, and then we have the data of the sequence data. So using the sequence data and the bioinformatics tools, we screen uh, the genes, actually the amino acid sequences, which can be used for the new antimicrobial peptides. So thereby, we found several candidate sequences. So after that, we characterize various properties uh, like a charge and hydrophobic residues, so on. And then we synthesize the synthetic peptide of these antimicrobial peptides and did the functional studies. And also we are focusing on these antimicrobial peptides to uh, encapsulate with the nanoparticles. Especially we focus on um, chitosan nanoparticle based encapsulation drug delivery system because sometimes these antimicrobial peptides are uh, having some problem when we give orally. It may digest with the enzymes. So we have to find some better way of therapeutic applications. Therefore, we encapsulate these antimicrobial peptides and compare the efficacy between the normal antimicrobial peptides and encapsulated peptides. And then we apply patents and publications. So with this, we discovered 3 novel antimicrobial peptides over the last 1 year. So one is the octopromycin, second one is octomycin, third one is octoprohibitine. So these are the properties or the characteristic of antimicrobial peptides what we are working on. So you can see these peptides having a lower amino acid sequence, lower than 50. And also the secondary structure prediction we can do. So all three have the alpha helix structure, so we can check their net charge. All are positively charged, and hydrophobic ratio and the molecular weight is we can determine. And then we have checked the various type of activities. Usually we check the antibacterial activity and also antifungal activity and antiviral activity. So additionally, we check the anti-inflammatory or immune modulatory activities. So these are the tested activities with positive results. So I want to just show you some of these results. So we do the time kill kinetics study related with these antimicrobial peptides. Here one antimicrobial peptide, octopromycin, was selected, and we use Acinetobacter baumannii. As you know, it is highly resistant for various antibiotics. So we have this resistant strain. So we always use the resistant bacterial strains. Um, so here we can see that the once we treat with the this octopromycin, the highly damaged cell of the baumannii is so with MIC, or minimum inhibitory concentration, is 50 microgram per milli, and minimum bactericidal concentration 200 microgram per milli. And here we use the chloramphenicol as the positive control. So when we compare this positive control and the octopromycin at MBC level, it's almost similar. Or so we can say that this octopromycin has a strong antibacterial activity against the Acinetobacter baumannii. And to understand a mode of action, so what we can do is we can check the various uh, assays related to the change of the cell wall permeability or the ROS production in the cells. So here we use the PI uptake assay. The PI means the propydium iodide. Normally when the cell membrane is changed due to the cell death, the PI penetrate into the cells and then bind with the chromosomal DNA. So when we treat with the octopromycin, you can see the more red cells are in there. So with the increase in the concentration, the number of red cells are also increased. So it indicates that our treatment has increased the cell death in Acinetobacter baumannii. And also we can use the ROS analysis with the bacteria using the fluorescent dye. So when ROS is generated, that cell indicates the green fluorescent. So more ROS indicate the higher oxidative stress in the cell, and thereby it may damage to the DNA, or it can uh, inhibit the growth of the bacteria cells. And also we check the octopromycin whether it has LPS binding or lipopolysaccharide polysaccharide binding ability. So it binds with the LPS and also it degrades the DNA. We use two type of DNA, plasmid DNA and the genomic DNA. So with the increasing the peptide, so the DNA degradation is visible. So it indicates that once we treat the peptide, so it can degrade the bacterial DNA, and that could be the one of the reason for cell death. And also we check the anti-biofilm activity of this peptide. So it also has the anti-biofilm by biofilm inhibition and also the biofilm eradication. So these are two type of phenomenon. Inhibition is related to the not allowed to grow the bacteria. Here the eradication means that already formed biofilm is removed from the surface. And also with the treatment, the persister cell inhibition also observed. So thereby we confirm that octopromycin has antibiofilm activity. And there is another activity. Usually we can check with the quorum sensing related activity. So when we check with this peptide, so it can inhibit or reduce the allogeneic production, which is related with the quorum sensing function. Quorum sensing is a kind of a cell-cell communication uh, while making the biofilm. So it is related with the biofilm activity. So we confirm that it can disturb the quorum sensing and thereby reduce the formation of the biofilm. And also we then check the toxicity in vitro and in vivo. And then we found that in vitro level, it has IC50 around 500 microgram per milli. And also we check the hemolysis activity and also the in vivo toxicity with zebrafish embryo and larvae. So in our lab, usually we use the zebrafish model. And then after selecting the safety dose of the octopromycin, we did the challenge experiment with the Baumani, Acinetobacter baumani, and we found that with the treatment of octopromycin, we can uh, increase the relative percent survival of the challenged Baumani challenge zebrafish. So bacterial load in the the spleen and kidney also decreased with the octopromycin treatment. So overall, we could say that this type of peptide can be applicable to treat and further test with the uh, next steps. So as a summary, actually, uh, basically these multi-drug resistant pathogens are the main issue at the moment. So we have to develop some strategies to control these pathogens. So antimicrobial peptides are one of the potential candidates with various benefits. So with our experiment results, actually we explain our octopromycin. It can change the membrane permeability, and also it can induce the reactive oxygen species and thereby induce the oxidative stress in the cell, and also it may damage the DNA in the bacteria cell, and then also it may facilitated by the binding to the LPS. Also, this octopromycin can inhibit the biofilm formation and also reduce the increase the eradication of the biofilm. So thereby we suggest in our study this octopromycin has a potential to be further developed as a candidate material or the alternative to control the Acinetobacter baumani. So we are focusing to test the pharmaceutical related applications, especially the pharmacokinetic studies. And also we are doing some encapsulation of this product and compare the efficacy data. And also currently we work on to check the expression of the genes related to the antimicrobial resistance in Baumani with the uh, sequencing of the bacterial genes. So those are the things what we are conducting on that. So similar studies we have conducted for other two uh, peptides as well. So you can find all the details about these peptides, since we have published uh, 3 papers actually related to the one of the peptides. Other two peptides actually we applied the patent. So until get the approval, we are not able to publish the paper. So those data will be published hopefully by next year. So that's all about, and these are my lab members. This is my 11th year here. So currently we have one research professor and 2 PhD students and 3 master students. So that's about the lab. And just a final thing about from the WHO, and they advise everybody to minimize this problem, to get the advice of a healthcare professional before taking the antibiotics. So that I think that is very important message. So I just want to share at the end of this. So if you have any suggestions or any questions, I can answer at this time.
Yeah, thank you, Professor Mahanama. Yeah, thank you, Professor Mahanam, for informative presentation, and nicely you have shown us that the novel finding that you have reached out, and actually we are very proud of you because as being Sri Lankan residing in South Korea and having many number of research students, postgraduates and postdoctoral fellows with you in your research lab, I think we are proud with your research work. And I think we have one question, a couple of questions. I will pick you one that the question is, asking to explain the bioinformatics tool you have used to identify candidate of antimicrobial peptides from transcriptome.
Yeah. So in the case of transcriptome, we have a nucleotide sequence, and then we convert that into the predicted amino acid sequence. So it is publicly available. You can easily get that. There are many actually in NCBI also, you can find it, or there are many other labs, they provide this tool. So if you have the nucleotide sequence, so you can convert into the amino acid sequence. And then the most important thing is to know whether that amino acid sequence has any has potential to be antimicrobial peptide. So there is there are several actually. There is one of the main database we call antimicrobial peptide database.
Okay.
So if you go to there, so you can put your sequence and then analyze whether that sequence has a positive charge or there is any hydrophobic ratio, and what is the identity of your sequence with the known antimicrobial peptides, because they can match your sequence with the known or the available antimicrobial peptides. And also they give some prediction whether your peptide has chance to be a antimicrobial peptide. So say like if you have a 50 amino acid sequence, sometimes you may not get the uh, uh, possibility to be a antimicrobial peptide. So what you have to do, you have to select some part of your peptide and then analyze. So what we did actually, I have selected more than 50 sequences like that, and then part by part we analyzed and we checked the charge and hydrophobic ratio and the structure like that, and then we, based on that data, so we can synthesize the synthetic peptide. Usually we synthesize around 10 milligram, so that is more than enough to get the initial results related to the antimicrobial activity. So that's
Thank
how
you.
we did
Yes.
actually.
Thank you, Professor, for full answer. And there's another question. In future, is there any chance to that MDR pathogen can develop resistance against antimicrobial peptides?
Yes, yes. So it is known, but there are many reports about the resistant development against the antimicrobial peptide as well. But since antimicrobial peptides can have different mode of actions for the same bacteria, for example, it can damage to the cell wall or the cell membrane, or it can damage to the DNA. So bacteria is not efficient to develop the resistant fastly than against the antibiotics. So it can develop against the antimicrobial peptide, but very slowly.
Okay, thank you. There's another question. What is the sequencer which is used for Baumani, that is the bacterial pathogen, AMR gene sequencing?
Can you please repeat? Which
Yeah,
sequence?
it's asking what is the sequence that you have you used in the AMR gene sequencing?
AMR gene. What is AMR gene?
That is respective to that the bacteria that you have shown in the presentation.
Which slide?
It's not really mentioned that slide.
You want to know our peptide sequence?
Yeah, the question is asking what is the sequencer?
Yeah,
Yes.
antimicrobial peptide sequence, right?
Yes, yes.
Yeah. Right now, we can give only the octomycin sequence because it is only the published paper. Other two, actually, since we are waiting for the patent, so
Okay.
it is not available right now. But if somebody wants to get the octomycin sequence, they can get our paper, or if they want, I can send.
And there are a couple of keep on coming the expectations. Can I introduce?
Yes, yes.
Okay, what are the tools that you use for the predicting structures of the peptides?
We use I-test.
Okay. Especially asking about the antimicrobial resistant gene sequencing, right? Before the question. Okay. Next one.
No. Sorry.
That
Antimicrobial
resistant gene, that AMR means that meant by the question was gene sequence AMR antimicrobial resistance.
No. We didn't do the sequencing now. We have done whole sequencing of the bacteria, not individual genes. Previously, actually, I don't know whether is it the correct answer. What I understood actually, they want to know how we get the antimicrobial resistant genes from Baumani, right?
Yes, that's true.
Yeah,
that's correct.
Okay,
yes.
so there are several reports about those genes. So we can easily get those genes and design the primer. And then those primers we use when we need to analyze those genes. But normally it may not perfectly match. So then we have to, because our strain is not same like in others. So for that, that's why we have done the sequencing of whole transcriptome, but still we haven't get the results of our Baumani strain.
Okay, last question. Instead of transcriptome, can we use proteome directly?
Yeah, it is possible, but it is expensive. That's more expensive and little complicated, and that's why we want to do, but right now we haven't.
Okay, thank you. Then that's all the questions that we have in the chat box. Okay, I think we can come to the end of this webinar. Would you like to share anything, Professor? Because you are doing excellent work with there, and thank you for being participating and accepting our invitation. And I know you are very busy with that, research and other administrative things. So would you like to share anything, and then we can wrap up, and thank you for.
I don't have any specific thing, and thank you again for inviting. And what I can tell you that if somebody wants to get our help for any sort of experiments or anything, they can contact. We can help in that way. Also, some of the sample, if you have some, for example, system to test for, if you have a specific virus or antiviral assays, something like that, so we can arrange the sample and we can develop some collaboration. That type of things we can do.
Thank you, Professor. Thank you for everything.
Yeah,
and on behalf of the Institute of Biology, Sri Lanka, I think we have come to the 6th webinar talk organized this year. And thank you very much for being participating, all the audience. And thank you for everything. Good night. Have a nice day.
Yeah, good night. Thank you.